We'll start off with the passage on the immune system, then the passage on alcohols, and we'll do 2 more passages together on hormones and enzyme kinetics. Right. How did everyone find this passage? Easy, medium, difficult? Okay, that's good. So I'll show you what I highlighted. I usually say this in every practice session, but it's important to highlight only what you need. If you highlight too much, you'll get lost in your highlights and it'll be difficult to see what things are important. So the first thing I highlighted is the word development because this is what the researchers are going to study. And the next word I highlighted was birth and then adult. I figured that the age was a variable used in the experiments, which could be asked about later on. I then highlighted accepted because this is the key result of experiment 3. And finally, I highlighted shared a placenta because this is the key difference between experiment 4, like the 2 experiments within experiment 4. Any questions about my highlights? All right, would anyone like to volunteer their answer for the first question? All right, Brian, why do you say B?
Because the experiment 1 show up if there's like if they first getting compare experiment 1A with 1B. 1B is like half of the pituitary gland, which I would assume they have some kind of exposure to their tissue, so the group will not be rejected, and which will be similar to the instructions.
Yes, exactly. So you were correct. The answer is B. So if we look back at experiment 1, we see that if the entire pituitary gland is transplanted and given back to the donor, it's rejected. The only way that an original organ is later rejected is if the immune system isn't taught that the original organ is considered self. Before the frogs reach adulthood, so when they're developing, their immune system is instructed that this new pituitary gland is their own. So if a new one is introduced, even if it was originally theirs at birth, it will be rejected because the immune system didn't have time to be taught that the original gland is self. So I drew this diagram to help better explain what is happening in experiments 1A and 1B. In the top, you see the full gland in green that's transplanted and recognized by the immune system during development. So when the original gland in pink is transplanted back, the body doesn't recognize it and attacks it. But in the bottom diagram, only half is transplanted. So the immune system recognizes both the original gland in pink and the transplanted gland in green. So in adulthood, when the original half is transplanted back, the body is able to recognize it and won't reject it. So A is wrong because experiment 1A showed that the immune system rejected the gland in pink in adulthood, which is genetically identical to the organism. C is wrong because the immune system, because in experiment 1B, the frogs reached adulthood and their immunological self-tolerance was maintained because the tissue must be present during maturation to be tolerated. And D is wrong because the foreign graft wasn't rejected. It was originally there at birth, so we can't make conclusions about foreign grafts based on these experiments. All right. Any volunteers for question 2? All right, Brian, why do you say B again?
Because basically the experiment 4 is talking about the identical twins would be more easily to accept from some other half of the identical twins, which would be a phenomenon since they both have 100% same DNA.
So experiment 4 says that they're non-identical. So, okay, let's start with A. A is wrong because non-identical twins in the womb are genetically similar, so genetics can't be a factor. They don't share the same genes. If we're sticking with this argument, then B is also wrong. While one twin who shares a placenta with their other twin can have reduced blood flow, this wouldn't affect immunological self-tolerance, so D is wrong. Our answer would be C because somehow when twins share the same placenta and therefore the same blood supply, their immune system would support each other's because they are both exposed to the same antigens in utero. Does that make sense? Okay, great. All right, what about 3? Okay, why do you say C?
I did this by process of elimination
Okay,
because I think C is the least extreme one.
okay. All right, let's look into it. So you're correct that C is right. The T cells in a rat born without a thymus gland would not be able to mature, so they wouldn't know what to target and kill. In other words, their immune system is weakened, so they will accept all foreign grafts more easily than if the rat was born with a thymus gland. This is why in experiment 2A, the rat who was thymectomized at birth developed the wasting disease as a result of the weakened immune system. So A is wrong because, like I said, their T cells won't attack anything, so it can't reject anything foreign. B is wrong because normally grafts from relatives are tolerated, but because the rat's T cells don't work, it'll accept more than just grafts from relatives. So the word only makes option B incorrect. And D is wrong because, like I said, for option A, the rat's immune system won't reject anything. So finally, C is correct because the rat will accept more grafts than normal.
Is this question purely a content knowledge? I was hesitant for this one because I didn't see there is any direct evidence from the passage.
Yeah, you're correct. So this question, you have to use your content knowledge. This isn't exactly like, how do I say this, like strategic. You just have to be familiar with the fact that the thymus gland is the center where T cells go to mature and recognize self versus non-self. And without a thymus gland, the T cells won't know what is self or what is not self. So they just won't do anything. They won't reject anything. So it's kind of like content review and inference, if that makes sense. All right, and for question 4. Okay, why do you say C?
Because I assume the parent is the closest one. If they share a different placenta, then I will assume the most similar one should be the parent, since they are directly relative.
Okay, so the keyword here is identical. So identical twins share the same genes. So they share the exact same tissues which are present during the development of the immune system. So they will both share the same self-tolerance during adulthood. Because they share the same self-tolerance, when they reach adulthood, they will be able to accept each other's grafts, whether or not they shared a placenta during gestation. So in question 2, if you remember, the scenario included non-identical twins who shared the same placenta and accepted each other's grafts. This is different. In that case, they did not share the same tissue type, but they were exposed to the same antigens throughout development. In this case, the twins share the same tissue type, but have different blood supplies. Blood supply doesn't matter in this case because the twins are identical. So they would accept each other's grafts. They could technically accept grafts from their parents or relatives, but A is the more correct answer. Does that make sense?
Can you please explain like how do you extract the information from passage for this one? Can you
Right.
please repeat again?
So this question doesn't really have much to do with the passage. You just have to pay attention to the word identical. So they're throwing this word in identical because they're telling you that these twins share the exact same DNA, the exact same tissue types, everything. So because they share the same tissues, they will accept each other's grafts during adulthood because their immune system, their T cells recognize each other's tissues.
So that has nothing to do with experiment 4.
Experiment 4. No, this is just it doesn't really have anything to do with experiment 4. They could just be throwing this question in just to, like, I guess, trick you a little bit, because in this case, non-identical twins who shared a placenta accepted skin grafts. So they're trying to give you a scenario where you have identical twins who didn't share a placenta.
I see. Thank you.
And I'm sorry about D being the same answer choice. That was an error on my part. All right. So for question 5, what did you guys get? Okay, why did you say D?
I think for the first item, first Roman numeral is just a counter knowledge, and which recognition of the first one is pure counter knowledge, and the second item is basically the first sentence in the passage, and the last item. I think the last item is also similar to the item 1, which is mostly counter knowledge.
Okay, so let's begin with item 1. So item 1 is true, like you said. This is the purpose of the thymus gland, which is to teach T cells of self versus non-self. Item 2 is actually false. The experiments don't suggest that the immune system is crucial for physical growth. Experiment 2A involves the wasting disease, but this is because of infection from the lack of T cells. So although the immune system plays a factor, it does not directly help physical growth. So we can't conclude that item 2 is true. Item 3 is true, and experiment 3 shows this. The adult rat accepted the other rat's islet cells. Does that make sense? Yes, I would say that metabolic growth is unrelated to physical growth. Let's look back at the first. I'm sorry, where did you see the word metabolism?
Well, I thought the item 2 is correct. Is first because of the first sentence. They say they play the central role in development of the immune system.
Yeah, just the
And
development
then-
of the immune system, not like physical growth.
And then I saw if the experiment 2A.
Mhm. Right. So like I said, because this rat was thymectomized at birth, it doesn't have a thymus. So the T cells won't know what to attack, so they won't attack anything. So therefore, the rat will be vulnerable to infections. And because it keeps getting all these infections, it develops this wasting disease.
Oh, because and also poor appearance.
Yeah, poor appearance probably just means like the rat is like, it looks sick.
Okay. I was confused with like the physical growth.
Yeah, I understand. That's why I said that this like question, I thought that this question was a little bit tricky as well.
Thank you.
No problem. Will growth hormone play a role on physical development? Yes, it does. Growth hormone specifically acts on the muscles and the bones. All right, last question. Any volunteers? All right, so this question I also found to be a little tricky. We can eliminate, well, first of all, we can eliminate B because we know that it's the opposite that's true. Younger animals are always more immunologically flexible than adults because they're still developing. C is wrong because if the T cells were activated to destroy the foreign islet cells, then the rat wouldn't accept it. But experiment 3 shows that they did. We can't really conclude that D is correct because we only know that the graft was accepted in an adult rat. If we're iffy about this, we can look at A. So A is more reasonable because in general, T cells in the thymus can be destroyed if they are programmed to kill self antigens. In other words, a function of the immune system is to eliminate T cells that are aimed at self antigens. So this is to prevent autoimmune disorders. Knowing this, we can assume that T cells specific to the foreign graft were killed because the transplant took place in the thymus, the very place where self-selection occurs. So let's imagine this. The pancreatic islet cells were implanted into another rat's thymus. By placing these foreign islet cells in the thymus, the T cells that would attack the foreign islet cells would be destroyed because the thymus is telling the T cells, oh, these foreign pancreatic islet cells are fine. Destroy yourself if you're programmed to kill these islet cells. So I drew this diagram to help explain. On the left, we have a case where the pancreatic islet cells in blue are somewhere else in the body, not in the thymus. The T cells in light pink will develop in the thymus and destroy the pancreatic islet cells because they're not self. And on the right, we have the islet cells in the thymus, where the T cells are taught that it is considered self. So the T cells that were previously taught to destroy islet cells will self-destruct. And so the animal will reject these foreign cells. Does that make sense? Okay, great. All right, and with that, we can move on to the alcohols passage. Yeah, I know the passage was easy to read, but like the questions were a little bit tricky.
Hey, can you guys see this screen? Okay, how did you find this passage? Okay, great. So these are some of the things I highlighted, just knowing where the table was and what enzymes were used, what products were made, and did anyone have any questions on the passage itself? If not, we'll go on to the questions. Okay, for number 1, does anyone want to volunteer and explain what they got? Okay, I'll go over it then. So to find the volume of 1.58 moles, we would have to use the equation that they gave us and the grams of ethanol molar mass, and they also gave us density. So By using this, we can convert from moles to grams and use the density to convert to liters. Does anyone have any questions? And this is half, so that would just cancel. We would multiply 2 into 46, and that would give us around 92. Okay, what about number 2? Yeah, do you want to explain how you did it? So for question 1, the formula is not in the passage, but is implied. So we were given moles, and we should be able to convert from moles to molar mass using, hold up, let me go back. So we're given ethanol, we should be able to find the molar, we have the molar mass given, so we should be able to go from moles to molar mass to grams. And then we also have the density, which is grams per milliliter. So we can cancel out the grams and just get the milliliters, which is the volume. So the formula is not given, but we have to use the table that was provided. Okay, how would we find the percentage of carbon in ethanol? Okay, so this is also using the figure 1 ethanol, and we would just count how many carbons there are. So since there's 2 carbons, we would multiply by how many grams of carbon there are, which is 12. You can look at the periodic table for that. And then again, to go from moles to grams, we would do the molar mass, which is given around 46. So we're just putting how many carbon there are over the total molar mass of ethanol. Does that make sense? Y would be C. Okay, moving on. Can anyone explain number 3? What did you guys get for number 3? Okay, yep. So again, we use the figure 1 equation, and we go from the grams that are given. And then the molar mass is also given, and it happens to be the same or as the grams of sugar that was used in the fermentation reaction. So those would cancel off. Oh wait, sorry, it's not the same, but yes, it's around 10. And then we would convert using this equation from moles of the sugar to the moles of ethanol, and again, use the molar mass. Does anyone have any questions on how we would do this? Okay, moving on. Number 4. What did you guys get for this? Yeah, could you explain how you did it? Yeah, okay. So I'll go through how we can use the equation. So we would use the grams of ethanol that they gave us. And since we want to figure out grams of acetaldehyde, we would have to use the equation as well. So converting from grams of ethanol to moles, we know the molar mass. And then we use the equation, it's a 1-to-1 ratio. And then we have to calculate the grams of acetaldehyde, which was right here. So you would just use a periodic table and add up all those carbons and hydrogens and oxygen, and then you would get 44. And then since we want the percent yield is around 80%, we would multiply 0.8. And that is around 17.6. Are there any questions? Let me know if you are confused. Okay, last question. Which is not conserved? So what did you guys say for number 5? Yeah, Brian, do you want to explain how you did this?
Because according to the figure E, the alcohol formation reaction, this is basically turning from one molecule of glucose into 2 carbon dioxide into ethanol.
Yeah,
Which
that's
is
perfect.
Mhm.
Thank you. So the number of atoms is conserved and mass cannot be created or destroyed. And the charge also stays the same. So it's neutral. And the number of molecules, we go from 1 into 4. So that does not stay the same. Okay, so we're going to give you guys 2 more passages to do, and I'll put mine in the chat. And we'll give you 20 minutes to do both of those and check back in. Does anyone need another minute?
All right, how did everyone find this passage? Easy, medium, difficult? It was difficult. Yeah, I understand. So here are my highlights. I highlighted corticosteroids because that's what's being measured. And I highlighted hypophysis and adrenal gland because these two organs are being looked at. I highlighted the word involution because this is what's been previously observed, that when the hypophysis is removed, the adrenal gland shrinks. I then highlighted sham because this is the control experiment, so I could potentially be asked about this. Any volunteers for the first question? Because we only have 10 minutes left, you guys can just type the answer that you got on the chat and I'll go ahead and explain them. Okay, you are correct that D is the right answer. The adrenal gland can be split into the adrenal medulla and adrenal cortex. The adrenal medulla releases epinephrine and norepinephrine, and the adrenal cortex secretes cortisol and aldosterone. A and C are wrong because aldosterone is responsible for the retention of sodium ions by the blood, which causes blood volume to increase. B is wrong because cortisol increases glucose levels in the blood and increases glycogenolysis in the liver to store glucose in an easily accessible form. D is the only thing that the adrenal gland doesn't do. Plasma calcium is regulated by calcitonin, which is produced by the thyroid gland, and parathyroid hormone, which is produced by the parathyroid gland. Right, what about this question? Oh, I'm sorry, before we move on to this one, did anyone have any questions about number one? Okay. So what did you guys get for this one? Yeah, so this one is pretty much straightforward. Like I said, this sham operation would be our control variable. The word sham means false. Like the passage states, the sham operation was just an operation in which no organs were taken out. So in theory, nothing should change. This operation just serves to prove that any changes in hormones are due to the removal of the organs, not because of the operation itself. And question 3. Yeah. So this question is also pretty much straightforward. Corticosteroids are derived from cholesterol, which are lipid soluble and are able to cross the plasma membrane. When steroid hormones cross the membrane, they bind to proteins to form the steroid receptor complex. This complex travels to the nucleus to induce transcription changes. Any questions about this one? Okay. And what about question 4? Yeah. So this question is just asking where aldosterone acts. It doesn't matter that the adrenal gland is missing because this is where aldosterone is made. But since you were injecting aldosterone, it will automatically travel to the kidney to induce changes in sodium and blood volume. Any questions about that one? Okay. And what did you guys get for question 5? Yeah, so the answer is A. The group 2 dogs lack an adrenal gland. The adrenal gland produces cortisol in response to rising levels of ACTH made from the anterior pituitary. Because these dogs don't have an adrenal gland, they won't make cortisol. The anterior pituitary will continue making ACTH until it sees cortisol levels go up due to feedback inhibition. So if cortisol never goes up, ACTH will continue to be high. So A would be our answer. C is wrong because it doesn't take into account feedback inhibition, and D is incorrect. ACTH levels depend on cortisol levels, which are products of the adrenal gland. Does that make sense? Any questions about this? Okay. All right, last question. What did you guys get? Okay, you're correct again. So ACTH is secreted by the anterior pituitary, which causes increased aldosterone and glucocorticoid release. So A and B are wrong. As aldosterone and cortisol increase, they will negatively feedback onto the anterior pituitary to secrete less ACTH. So C is wrong. Epinephrine and norepinephrine are secreted by the adrenal medulla and are not under the influence of ACTH or hormones secreted by the anterior pituitary. They are mostly stimulated by changes in the sympathetic nervous system. So our answer is D.
Quick question.
Yeah.
So for the last question, we basically just determine which hormone is regulated by the adrenal or anterior or posterior pituitary, and then we basically just using the flat peg to find the answer.
Yeah. Oh my gosh, I'm so sorry. I'll mute myself for just 2 seconds.
Mhm.
Okay, I'm so sorry about that. So so this question is asking you to select one of the options that the anterior pituitary gland does not do. So the anterior pituitary gland does not result in increased secretion of epinephrine and norepinephrine because, like I said, these two hormones are mostly stimulated by the sympathetic nervous system. So like aldosterone and glucocorticoids are dependent on ACTH, which is secreted by the anterior pituitary gland. Does that make sense? Okay. Oh God. Okay, Sim, you can take over now. Sorry about that.
Okay, thank you, Ashley. What did you guys get for number 1? Yeah, that's great. So it mentioned it in the passage. It also mentioned adding methyl groups, and transferases move the groups from one molecule to another. So if you didn't know that transferases do this, you still had some clues in the passage. I would definitely know these type of enzymes and what they do though, and specific examples. What about number 2? Okay, yeah, so if we're increasing the concentration of this cofactor, that's the reactant, and that's going to increase the rate, and by increasing the rate, we're going to get closer to Vmax. Okay, what about 3? Okay, so this is saying what would happen if we don't have the enzyme. So enzymes help the reaction because they lower the activation energy, and that speeds up the reaction. However, the reaction can still occur, but it's just going to take longer. So as long as the reaction is going to be spontaneous, it will occur, but at a slower rate without the enzyme. Okay, number 4. Yeah, sorry about that. So the correct answer is D. Yes. So if we're decreasing the cofactor, which allows methylation, that's going to decrease the amount of methylation on histone X, which would decrease gene Y, and that would decrease protein Z. Does anyone have any questions on that? No? Okay, perfect. So number 5, last question. And yeah, I'll go over this quickly. This was just the reversal of number 4, so saying the same thing. So instead, if we have histone methyltransferases, gene Y would increase, which would increase protein Z. Any questions on this passage? Okay, if not, thank you for coming to today's session. And if you have any questions, feel free to email us, and please donate to MCAL 101. Since we're all volunteers, we would really appreciate that. Thank you so much. And Ashley, do you have anything else to add?
No, I don't. Thank you guys for coming today. Sorry about the interruption.